Okay, Assalamualaikum and good morning to all of you. So we proceed with the chapter 7 known as the Basic Chromatography. Okay, the learning outcome for this topic where at the end of this chapter you should be able to explain about the principle of chromatography and types of chromatography on the basis of interaction of the analyte with the stationary phase and types of chromatography and technique, which is you can differentiate between planar chromatography and column chromatography, and also the applications of planar and column chromatography. So we proceed with the principle of chromatography first. Samples to be analyzed are usually a mixture of many components in a complex matrix. For sample containing unknown compounds, the component must be separated from each other so that each individual component can be identified by other analytical methods. The third one, measurement technique used for chemical analysis are usually specific for a single chemical species. A mixture can be separated using the differences in physical or chemical properties of the individual components. Example of physical properties that are useful for separation are density and size. Some useful chemical properties by which compound can be separated are solubility, boiling point, and vapor pressure. For classical separation method, so we have a masking, precipitation, distillation, solvent extraction, and also ion exchange, where this is a classical separation method. So at the same time, you can separate two of our components by using distillation and precipitation that you have learned in chapter 6. And then chromatography is, for example, is electrophoresis, which is it involves the separation between electron. So if let's say you have the positively charged of ion, so it will separate into the positively charged, where the negatively charged, or vice versa. So about chromatography, actually is a technique used to separate the component mixture based on the rates at which they are carried through a stationary phase by a gaseous or liquid phase known as the mobile phase. So for mobile phase is just only have two phase, which is either gaseous or liquid phase. Same for your analyte, you kena follow lah you punya nature of you punya stationary phase or liquid phase. Okay, your mobile phase yang akan bawa you punya sample tu untuk bergerak. Okay, stationary phase ni tempat untuk berlakunya separation. But both of stationary phase and mobile phase will play important roles in order to separate this component, this mixture into the components. So by using the illustration of chromatography, as you can see from the diagram, so you have the mixture, ya ada warna kuning, merah, biru, hitam, and then dia akan diseparatekan. Okay, we dip this, contohnya kalau chromatography kita rendamkan dia punya TLC plate, so dia akan mix, dia akan separate into rates. So yang nampak kat sini, yang kuning akan travel lebih jauh berbanding merah, berbanding hitam, dan berbanding biru. So biru adalah less travel lah. Tapi kalau ikut kat situ, kalau tengok kat situ, boleh tengok bahawa kita kena tengok bahawa yang kuning adalah nature, dia mempunyai nature yang hampir sama dengan mobile phase punya karakteristik. Sebab tu dia travel lebih jauh. Merah adalah less sikit, while hitam adalah very least. All types of chromatography contain a stationary phase with either solid or liquid to absorb or adsorb the mixture being separated, and a mobile phase, as I mentioned earlier, liquid or gas, which passes over the stationary phase and competes with it for the constituent of the mixture. Okay, as I mentioned, there is two phases, important phase, two phases, which is stationary phase and the mobile phase. So the uses of chromatography, Okay, actually to analyze, to identify, to purify and also to quantify. That's why chromatography ni banyak sangat diguna pakai, tak kisah di industri apa pun memang ada banyak. Walaupun you kata petroleum, dalam petroleum kita guna gas chromatography untuk kita purifykan kita punya components, and then kita nak quantifykan dia punya percentage of, contoh kita nak hasilkan you nak hasilkan ethanol, ethanol yang high percentage of ethanol dalam mixture tersebut. So you guna purify. So menggunakan alat ni kita boleh guna pakai. So for analyze, examine a mixture, its component and their relation to one another. For identify, determine the identity of mixture or component based on known components. Purify, separate components in order to isolate one of interest for further study. And last one is quantify. Determine the amount of the mixture and or the component present in the sample. This is a basic principle of chromatography. Samples, well, first one is a sample is dissolved in a solvent and washed through a stationary phase by mobile phase called as a eluent. Eluent itu adalah sebagai mobile phase, adalah mobile phase, and then competition between sample molecule absorbed by the stationary phase and dissolved by the eluent results in separations. Speed of the substance passes through depend on the nature of the stationary phase and the eluent. So meaning that, as I mentioned, either this time you need to focus more on polarity of stationary phase and also the eluent or stationary mobile phase. So important part is polarity ni depends. If let's say both of them is polar, meaning that there is no reaction will occur. So meaning that you need to have one of the stationary phase must be polar, the other one must be less polar or non-polar, baru reaction tu berlaku. Substances that are more soluble in the eluent and less absorbed by the stationary phase move faster through the apparatus. Ni macam tadi dalam gambar rajah tadi, warna contoh warna kuning tadi lah. So dia kata less absorbed pada stationary phase, so dia akan move faster through the apparatus sebab dia mempunyai nature yang hampir sama dengan mobile phase. Not exactly 100%, dia mempunyai karakteristik. Kalau awak punya mobile phase tu adalah non-polar, so meaning that you punya light tu pun warna kuning tu adalah non-polar. So tapi dia bukan benda yang sama, tapi dia sifatnya non-polar. Okay, components in the mixture separate out as the mobile phase move through the stationary phase. Itu yang paling penting. Okay, now we proceed with the principal chromatography separation. So as you can see from this diagram, okay, the sample is placed on the top of the column, but before that you need to fill the column with the stationary phase, right? And then you add a mobile phase in order to make sure the separation will occur, and then it will give the mobility of the sample you analyze. So as you can see from this, the individual component interact with the stationary phase and also the mobile phase to different degrees. Component A interact more strongly to the stationary phase. That's why dia suka dengan stationary phase, so dia akan stay lama dengan stationary phase tersebut. Okay, and then component B and component C is less interact lah. B and C tu less interact. So dia, tapi component A ni komponen B and C ni akan travel lebih jauh, lagi cepat lah. So they elute first and second. Alright, so we proceed with this slide. So retention time taken for a component in the sample to pass through the column, because you will use a lot of retention time terms in this topic. Okay, and then chromatogram ni biasa kita boleh guna pakai dalam gas chromatography lah. Maksudnya gas chromatography and also liquid chromatography, it will produce a visual output of the chromatograph, atau kita panggil sebagai chromatograph lah, which is it will show the different peak or pattern on the chromatogram, which is it will correspond to different component of the separated mixture. So for example, if let's say you can see here, the band 1 will elute first, component that elute first, because dia akan appear pertama sekali, dia yang keluar paling awal, because you punya graph ni adalah dia punya band 1 dengan pada x-axis tu adalah masa, time. So it shows that band 1 will represent component A, for example, in this case component A, meanwhile the second band or graph tu akan menunjukkan kepada component B, so which is really different time, so later daripada compared to band 1. So this is known as chromatogram. Y-axis is retention time, meanwhile x-axis is retention time. Y-axis is signal corresponding to the response created by the analytes existing in the system. So we proceed to types of chromatography on the basis of interaction of the analyte with a tertiary phase. As you can see from this diagram, the both left and right hand side. Okay, so there is many types of chromatography. Okay, of the analyte with a tertiary phase. So normally we will use adsorption. Adsorption is terjerap. Alright, so partition, ion exchange, and also size exclusion. For adsorption, so adsorption ni dia akan terjerap pada permukaan, solid absorbed on surface of tertiary phase only. So dia just absorb pada permukaan surface tertiary phase sahaja. Okay, yang dot-dot hitam tu adalah awak punya analyte, black color tu adalah analyte awak, so dia akan absorb kat situ. Tapi partition ni solute dissolve in liquid phase coated on surface of solid support, which is dekat solid support sama, dia punya gambar aja lebih kurang sama, tetapi di dalam awak punya bila partition ni dia ada layer yang lain yang membuatkan reaction berlaku kat situ. So you punya analyte awak akan lepak lah sekejap kat situ, atau untuk stay dia akan ada interaction dengan tertiary phase tu baru dia boleh bergerak. Okay, so we proceed with the first one, adsorption chromatography. Okay, a chromatography where the analytes component are separated based on their different degree of adsorption stick on a surface of solid tertiary phase. So in the adsorption process, the solute molecule are held on or attached on the surface of the tertiary phase, which is solid. The separation of solute depends on the difference in polarity. So polarity ni kita akan tengok sama ada dia polar, non-polar atau less polar. Okay, if polar in chemical structure, it contains of hydroxy group. Normally bila kita sebut polar ni ada contains of hydroxy group, OH group, or content of H2O, ataupun yang ada lone pair. Kita tahu kan macam mana nak tentukan compound tu adalah polar atau non-polar. You belajar dalam section 138, which is you ada belajar Lucas-Lewis structure. Kalau tak tahu Lucas-Lewis structure, then dalam organic chemistry pun sama juga, ada belajar kan which compound is less polar, non-polar ataupun polar. Okay, because of the different degree of intermolecular attraction, some component of the mixture will be more attracted or absorbed to the tertiary phase, thus separated. But depends, once again, depends on the types of tertiary phase, the polarity of tertiary phase, and also the polarity of the mobile phase. So what happened here? Absorption and absorption. So you need to know about this. Molecule adhere to a surface of the material. That one is we call as adsorption. For absorption, molecule are drawn in the material. Substances that are strongly absorbed move through a tertiary phase much more slowly than those that are weakly absorbed. The tertiary phase is a polar solid. Okay, in this case, the tertiary phase is a polar solid, usually finely divided silica and alumina, and the solute are more molecules. So material untuk buat tertiary phase adalah alumina and silica, and classified as a polar tertiary phase. So meaning that if this adsorption chromatography already stated that the tertiary phase is polar, your mobile phase must be less polar ataupun non-polar mobile phase, baru boleh guna pakai. Kalau tak berlaku reaction, tak akan berlaku separation. The separation of the solute depends on the difference in their polarity. The more polar solute are more readily absorbed than the less polar solute in the tertiary phase. Okay, mobile phase may be liquid or in a gas state. Boleh dua-dua ni. Okay, so once again, this is the absorption chromatography. As you can see from this diagram, okay, so the phase solid, okay, is polar, already polar. So mobile phase awak adalah liquid, solid awak adalah analyte. Mobile phase tu yang kawasan putih tu, itu adalah mobile phase dalam bentuk liquid lah. Dalam ni gambar rajah ni saya just tunjukkan, kita nak tahu yang mobile phase awak dalam warna putih tu ya. Surface phase awak ada kelabu, yang you punya analyte atau solid awak adalah yang warna hitam. So meaning that yang berlaku absorption kepada permukaan you punya surface phase adalah polar component, meanwhile yang tidak, warna hitam yang tidak melekat kepada ataupun tidak terjerap kepada permukaan surface phase awak tu adalah less polar component. Alright, sebab dia tak suka dengan surface phase, so dia suka dengan bawah phase, so dia akan hanging dengan bawah phase saja. Okay, we proceed with the partition chromatography. Okay, when we talk about partition chromatography, it is involved the partitioning is the distribution of the component between two immiscible phases. Immiscible phases meaning that dia tidak serasi ataupun tidak sama phases lah, ataupun dia tidak mix together dengan baik. Okay, separation of the component will be based on relative solubility of the components in the mobile phase and stationary phase. Also known as a liquid-liquid chromatography, the stationary phase is liquid supported on an inert solid surface of packing particle. Mobile phase may be liquid or gas. Separation is based on solid partitioning between two liquid phase. Okay, so you can see from this partition chromatography diagram, the stationary phase liquid supported on solid. Dia lekat, warna purple tu dia terlekat dan lekat dia punya, kita punya solid, supported solid untuk dipegang kat situ. So solid awak yang warna biru, so you punya mobile phase warna kuning. So tapi dia dalam bentuk bulat ni lah, ni nampak bulat ni, ni adalah bentuk macam pipe, getah pipe tu, dia panjang sepanjang tu. Kalau ikutkan dia punya, this is a cross section of open tubular column. So kalau you google partition chromatography ni, biasa bagi pedawat HPLC dalam gambar rajah ni. Okay, example, we have the partition chromatography, high performance liquid chromatography, very expensive instrument and very high tech instrument. Paper chromatography, gas chromatography as example partition. Okay, so you can see HPLC dia bentuk petak panjangnya ni. So sebenarnya dalam tu ada kolom. Nah, kolom ni macam ni. Ni GC pun ada kolom. Okay, kolom dia berbentuk gulung dia bergulung macam getah tu, tapi dia punya layer tu banyak. So untuk berlaku separation dalam tu. Paper chromatography also same, dikenal sebagai partition chromatography. Okay, there is a two main type of partition chromatography, which is normal phase liquid chromatography and reverse phase liquid chromatography. So actually normal phase ni bila saturated phase awak polar, mestilah awak punya mobile phase adalah non-polar. Kalau reverse terbaliklah, saturated phase non-polar, mobile phase adalah polar. Okay, sebenarnya bila guna partition chromatography ni untuk guna high tech instrument, for example gas chromatography and also high performance liquid chromatography, kita boleh tukar dia punya polarity of the tertiary phase sebab kita punya nature of samples or analyte of solid awak adalah berbeza-beza kan polaritinya. So kalau kita fixkan saja dia punya normal phase chromatography, polar tertiary phase and non-polar mobile phase, susahlah kita nak buat separation yang lain kan. So that's why dia ada beberapa, ada baguslah dia boleh tukarkan benda ni. Okay, so this one you can read by your own. Okay, type of chromatography and technique. So we have a planar chromatography and column chromatography. So saya dah simplifiedkan dalam table ni type of chromatography, which is absorption chromatography, type of tertiary phase, type of mobile phase, and example. Kita kena pakai. So for absorption chromatography, solid, dia punya tertiary phase is solid, either alumina, cellulose, or silica. So you find out the alumina structure, cellulose structure. So all of these contain of OH group. So that's why they are polar component. So that is how you nak identify the polarity of that component. Mobile phase liquid solvent such as alcohol. Alcohol ada OH group tapi dia less polar than alumina ataupun cellulose. Sodium chloride and also gas boleh dipakai. Example column chromatography, TLC. and also gas solid chromatography. Partition chromatography, absorbent liquid on solid surface, liquid solvent, gas such as helium. Bawah phase dia. Paper chromatography, gas liquid chromatography. Okay, yang itu boleh pakai. And then this one, column and planar chromatography, type, tertiary phase and mobile phase. So for column or elution chromatography, tertiary phase dia adalah inert solid such as aluminium oxide, silica placed in a narrow tube or column. Mobile phase liquid solvent such as benzene. Planar chromatography, either paper chromatography, PC, saya abbreviation dia PC, thin layer chromatography. PC water absorbed on the paper, so dia ada permukaan itu ada layer, dia ada tertiary phase yang solid support, dekat layer atas dia ada water absorbed on the paper. Itu paper chromatography. So TLC, a solid absorbed such as alumina, solid gel. And then you can learn, after this you will learn more about the TLC. Mobile phase, either PC and organic solvent, TLC a solvent juga lah, liquid. So this is an example of the you punya senior dulu sebelum PKP kita buat. So this is a paper chromatography punya, kita punya apa experiment. So you have the mixture containing of this one, yang first one mixture, and then the second one you dapat blue, green, red and yellow. They separate into 4 color from the mixture. So yang biru ni macam mana kita dapat? Kalau bulat ni seluruhan, you kena gariskan, ambil pada hujung dan hujung, lepas tu bahagikan 2, baru you boleh kira ketinggian dia berapa cm. So sample travel cm over solvent front travel. So you will have the RF value for retention time for blue color. Okay, so this one lah, draw the function. You need to use a pencil to draw a line because it is made up of carbon and are not involved in separation. Okay, this is an example of question from chapter 7. Question 8 ni, explain the difference of tertiary phase between a planar chromatography and column chromatography. And then a column chromatography was set up using water coated on solid support as the tertiary phase and organic solvent as the mobile phase. Indicate how two compound of different polarity can be separated through this column. So saya nak berfikir untuk 3 soalan ni. TLC is used in separation of mixture of 2 components X and Y. X is higher polarity than Y. By using polar solvent, explain the principle of separation using a TLC. Identify the components that are left behind at baseline of the compound. Okay, so that's all. You need to give a try to answer all these questions, or later kita akan discuss. That's all. Thank you.